Hello everyone, I am Ginika Uzoekwe, and I'll be presenting on the topic frequency and activation status of immune markers in human whole blood. Let's get this out of the way. Okay. So on this slide, I've brought the diagram of a cell line. This is a stem cell. These are the different subsets, but I want to particularly show the T lymphocyte because that's going to be the basis of the presentation today. The T lymphocyte has two different subsets, the CD4 T cells and the CD8 T cells. These are really important for immune function. You would agree with me that collection, processing, and preservation of immune cells are still in its infancy when compared to collection, processing, and preservation of other biospecimens like the DNA and the serum. The DNA and serum has a more standard procedure for processing and preservation. Yeah. That being said, for any kind of immune cell analysis, be it the response pattern, be it the immune levels, investigating immune levels, be it investigating clinical relevance of newly discovered biomarkers, we would and always require uncompromised blood samples, which will allow for the isolation of viable and functionally competent immune cells. Let's move this out of the way. Okay. So developing countries face challenges with sample processing and preservation. There's also difficulty in transport to central facilities. Yeah, so some countries, especially in Africa, where samples have to be transported to the site of collection to probably the cities which have the central facilities for processing. Now, the time lapse between blood sample collection and the actual test analysis may have an influence on the frequency. That's when I say frequency, I mean the number of viable cells, and then the activation status of the T cells. And another factor that might have an impact on these two parameters another factor that might have an impact is the type of collection tube used. Now, there are different collection tubes in the market, and we know that the different collection tubes have different chemicals in them that use different mechanisms for preventing, inhibiting coagulation of blood. Next slide. Okay. The aim of the study, therefore, was to determine the frequency, expression frequency, and diffusion status of T cell at different study time points and a different blood collection tube. Basically, we wanted to know if time difference, that's the time lapse between actual blood collection and actual analysis, has any impact. Are some of the cells getting killed or non-viable because of the difference in time lapse? Because if this is so, it's going to affect any analysis that we done on the immune cells. That's going to lead to false results. And also, does the time lapse have an effect on the activation status? Because if we're going to be doing probably something like a response pattern analysis on the immune cells, and then a time lapse on its own affected the activation status, then our results won't basically show the response pattern to whatever activation we're going to use, but it's going to be a false result. Yes. And another thing we would like to investigate here also is the different blood collection tube. Is the difference in the chemical in this different blood collection tube having any does difference in the different blood collection sorry does the chemicals in the different blood collection tube have any effect on the expression frequency and the activation status of the T cells? That's another point. So specifically, we'd want to determine the frequency and the activation status of T cells using anti-human antibody CD4, CD8, CD69 at the various time points. Now, the CD4 antibody would bind to the CD4 markers on the CD4 T cells. It's like the ELISA principle. Also with the CD8. And now for the CD69, normally when T cells are activated, they express a particular molecule on their cell surface. The CD69 molecule is an early activation marker in this T lymphocytes. So the anti-CD69 will basically bind to any cell that is activated. And we're analyzing at different time points. Time less than 1, this is like simulating analysis just after blood sample collection. So we did the analysis within 1 hour of blood sample collection, and the next time point was 4 hours. So simulating traveling from a particular region to a central lab that would last for where the journey would last for 4 hours. So we want to see how the frequency and activation status differs when compared to the analysis done just after blood sample collection. And again, to determine the frequency and activation status of T cells collected with different preservation tube. So here again, we want to know if the different preservation tube would have any effect on the frequency and activation status of the T cell. The finding of the study will be important when selecting an optimal time point and collection tube for T cell analysis. This information will also assist researchers as to whether or not to source for an alternative method of processing, especially in rural community. Because if by the end of the project we realize that time lapse has an effect, or a particular collection tube has an effect, yeah, we would make decisions based on these results as to whether to change, because basically it would affect our T cell analysis. Ethical approval was obtained from the Committee of Human Research Publication and Ethics at the School of Medical Sciences, Kwame University of Science and Technology, Kumasi, Ghana. 5 ml of venous blood was taken from all 9 participants into EDT and heparin tubes, and the whole blood staining using anti-human specific antibodies was performed on the samples using a standard procedure. CD3 APC. So here basically the APC, the Alexa 488, the PRCP, and so they are dyes that was bounded to the antibody. This is just like the ELISA technique, immunochemistry, yes. So the stem cells were analyzed using the fluorescent activated cell sorter. The fluorescent activated, just as the name implies, is sort cells. The fact is a subset of the flow cytometer. This is a high throughput technological equipment that can simultaneously detect and quantify the physical and chemical properties of a population of cells. Now the data obtained was analyzed using the graph path Prism, and independent t-test was used to compare the data groups. So this is a flowchart of the major processes for this particular project. So first of all, there was blood sample collection, there was whole blood staining. This is me doing the whole blood staining. Then there was FACS analysis. We used the BD Acuri type of the facts to analyze our samples. And this is a monitor showing the results, the interpretation of whatever that was going on in here. And basically here I was trying to get on a particular population of my desired cell. So here we had the gating strategy and the data analysis here. For the results and discussion, first of all, we investigated the impact of time on the frequency of CD4 and CD8 T cells. So for figure 1, this showed the CD4 T cell expression in EDTA, CD4 T cell expression in heparin, CD8 T cell expression in EDTA, CD8 T cell expression in heparin. So we wanted to know if the expression that's the frequency, the number of the T cell, if it had if it changed in just after 4 hours. There was any change in the frequency. So was there less viable cells? Were the cells dying as a result of the time lapse? Yes. So since we analyzed that p-value less than 0.05, this wasn't significant. 0.755 wasn't significant as well. 0.185 wasn't significant, and we could conclude, since there was no significant change in the frequency of the CD4 and CD8 T cell after 4 hours time lapse, we could conclude that there's no impact. Time has no impact on the frequency of CD4 and CD8 T cells. And when I say time, I mean the 4 hours interval we used. Yes. And here we investigate the impact of time on the activation of CD4 and CD8 T cells. Earlier I told you that we measured activation by the anti-CD4 CD69 antibody, early activation marker. Yes, and here we compare the CD4 T cell activation in EDTA, CD4 T cell activation in heparin, and also the same with CD8 T cell EDTA and heparin. And right here, we saw that there was a significant difference in the activation status of the CD4 T cells, and also there was a significant difference in the activation status of CD4 T cells right here. Well, let's notice an important trend here. The difference in activation after 4 hours happened only in the T cells that were placed in the EDTA tubes. You can see here, but right here in the heparin tubes, there was no such significant difference. So this may point that it was not only the time lapse of 4 hours that caused the activation. Probably the EDTA tube had some effect on the change in the activation status after 4 hours. Right here, we moved on to investigate the impact of collection tube on the frequency of CD4 and CD8 T cell. And the figure 9 shows the CD4 T cell expression at time 0. That's sometimes I put time less than 1 hour, but it just shows analysis just after blood sample collection, so there was no time lapse in this place. And here too, CD4 T cell expression at 4 hours. That's after 4 hours of blood sample collection. Again, CD8 T cells time 0 and 4 hours after. And here we can see that the type of collection tube, be it EDTA or heparin, had no effect whatsoever, because 0.89, 0.77, 0.5, 0.6. So the collection tubes used had no impact on the frequency, that's the number of CD4 and CD8 T cell. Next, we investigate the impact of collection tube on the activation of CD4 and CD8 T cell. So CD8 T cell activation at time 0, CD4 T cell activation after 4 hours, CD8 T cell activation at time 0, CD8 T cell after 4 hours. And here we could see that there was a significant change in the activation status of the CD4 T cell at time 0. And also there was a activation change here as well. So for both the CD4 and the CD8 T cell, there was activation at time 0. So here we conclude and say that time has no impact on the expression frequency, but may have an impact on the activation status of T cells. And also, EDTA and heparin collection tube used has no impact on the expression frequency, but may have an impact on the activation status. So basically, for both the time lapse of 4 hours and type of collection tube, the number of the T cell weren't significantly affected. The number, expression frequency, they all mean the same thing, weren't significantly affected. But on the other hand, the activation status of these T cells, of both CD4 and CD8 T cells, were affected both by time lapse of 4 hours and the type of collection tube used. Yeah, and from right from here, we could see that the 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 impact happened in the EDTA tube, and in fact, this result has been supported by other research carried out, and they proposed that the heparin collection tube may be a more stable collection tube for T cell analysis than the EDTA. Yeah. Okay. So we recommend that the impact of other time points and other collection tubes like the citrate on the expression pattern and the activation frequency of T cells should also be investigated. So we may want to know what would happen, how would a time lapse of say 6 hours, 8 hours, 12 hours, 24 hours after blood collection, how would how would the time lapse affect the frequency pattern and the activation status of these T cells? And also, we would also want to know how other collection tubes like citrate would also impact expression frequency and activation status in this case. These are my references, and thank you. Enjoy the rest of the conference.